Hi friends, my name is Dev and today I am going to discuss about glycolysis. Glycolysis is a major metabolic pathway that utilizes glucose, which is a digestion product from the carbohydrates, and this glucose is being converted to pyruvic acid through the process or through the pathway of glycolysis. Okay, here into the glycolysis, the 6 carbon compound glucose is being converted into 3 carbon compound pyruvate, and in the during the process, it also liberates 2 ATP molecules and 2 NADH molecules. And overall, the glycolysis process occurs into the cytosol of a cell and is essentially anaerobic. That means into the process of glycolysis, no oxygen is involved. Now we'll start. This was the introduction about the glycolysis, and now we will start with the pathway. There are two phases into the pathway of glycolysis. One of the first of which is preparatory phase. Okay, so from reaction number 1 to reaction number 5 of the glycolysis will be talking about the preparatory phase, in which energy will be required. That means there will be expenditure of energy to add phosphate groups to glucose, and also the glucose will be converted to the 2,3 carbon molecules dihydroxyacetone phosphate and etheraldehyde 3 phosphate. So we will start with the preparatory phase. Into the preparatory phase, the first reaction is the phosphorylation reaction, in which a phosphate group from the ATP, this phosphate group, a phosphate group from this ATP will be added to the glucose molecule on the 6th carbon position. So one phosphate group is coming from this ATP and is being transferred to the 6th carbon position of this glucose molecule by the action of the enzyme hexokinase. We all know what is the property of the hexokinase. Hexokinase or all the kinase enzymes. Kinases are those class of enzymes which transfers a phosphate group into a molecule of any compound. Okay. So here hexokinase is utilizing this transferring this phosphate group from ATP to this glucose molecule, and here the cofactor which is being utilized by this hexokinase is Mg plus plus. So Mg plus or Mg2 plus or Mg plus plus is being utilized by this enzyme hexokinase to transfer this phosphate group from this glucose to this glucose molecule on the 6th carbon position. Then comes a second reaction in which this glucose 6-phosphate is converted to fructose 6-phosphate. This is an isomerization reaction, and we know that fructose is the isomer of glucose, where glucose contains the aldehyde group and fructose contains the keto group. Here the enzyme phosphohexose isomerase is converting this glucose 6-phosphate to fructose 6-phosphate. The glucose 6-phosphate, the aldose from reaction number 1 undergoes isomerization to fructose 6-phosphate, which is a ketose. Okay, so here this aldehyde group of this glucose 6-phosphate is converting to the ketose group, and this reaction is catalyzed by phosphohexose isomerase. Okay, now we'll move on to the third reaction, which is again a phosphorylation step. Into this step, fructose 6-phosphate again accepts a phosphate molecule from this ATP, okay, and forming fructose 1,6-bisphosphate. Okay, here the enzyme involved is phosphofructokinase 1. Here this incoming phosphate group is added to the first carbon position of this fructose 6-phosphate, thereby making it as fructose 1,6-bisphosphate. So it is the second step into this preparatory phase, where we are requiring one molecule of ATP to transfer the phosphate group to this fructose 6-phosphate, forming fructose 1,6-bisphosphate. In the next step, which is the fourth step of this pathway, there occurs the cleavage of the 6 carbon sugar phosphate. Okay, this 6 carbon sugar phosphate is cleaved into 3 carbon sugar phosphates, which is first one is the glyceraldehyde 3 phosphate and second one is the dihydroxyacetone phosphate, which are both the isomers. So glyceraldehyde 3 phosphate and dihydroxyacetone phosphate is being produced from the fructose 1,6 phosphate by the action of enzyme aldolase. Okay, now comes the final step of this preparatory phase, which is the conversion of dihydroxyacetone phosphate to glyceraldehyde 3 phosphate. So we are ultimately getting 2 molecules of glyceraldehyde 3 phosphate. So here the enzyme triose phosphate isomerase, as I told you, is glyceraldehyde 3 phosphate is an isomer of dihydroxyacetone phosphate. So here also the isomerization is happening and this reaction is catalyzed by the enzyme triose phosphate isomerase, which is cleaving this fructose 1,6 phosphate into 2 molecules, one of the glyceraldehyde 3 phosphate and another of the dihydroxyacetone phosphate. Glyceraldehyde 3 phosphate contains an aldehyde group and dihydroxyacetone phosphate contains a keto group. So here the blue shaded region is the aldehyde group containing glyceraldehyde 3 phosphate and this pink shaded compound is the dihydroxyacetone phosphate. With this reaction number 5, the preparatory phase is complete, and now we will move on to the payoff phase. Payoff phase we can also call as energy production phase, because here we will be getting a net gain of 2 ATP. We have utilized 2 ATP into the payoff phase, and here we will be getting 4 ATP. So there will be a net gain of 2 ATP, and also 2 NADH will be produced into the reaction number 6. So ultimately we are getting 2 ATP and 2 NADH. Okay, so this payoff phase starts with reaction number 6, which is the oxidation and phosphorylation step. That means here both oxidation of this glyceraldehyde 3 phosphate to a glycerate group is occurring. So this glyceraldehyde is converted to glycerate group. So here the oxidation of this glyceraldehyde 3 phosphate will lead to the formation of 3 phosphoglycerate, and this oxidation here glyceraldehyde 3 phosphate will be oxidized to form 3 phosphoglycerate, releasing protons that will be accepted by the 2 NAD plus and forming 2 NADH and H plus. So here this NAD plus is accepting the protons released by glyceraldehyde 3 phosphate and is forming 2 NADH plus H plus. But as I told you that this is the oxidation and phosphorylation step, so it is not directly forming 3 phosphoglycerate, but also what is happening is the addition of a phosphate molecule to this glyceraldehyde 3 phosphate. Here glyceraldehyde 3 phosphate is oxidized to form 3 phosphoglycerate, and simultaneously addition of another phosphate molecule, inorganic phosphate molecule, is being done to form 1,3-bisphosphoglycerate, and the enzyme which is catalyzing this step is glyceraldehyde 3 phosphate dehydrogenase. Now into the next step, which is the 7th step, there will be formation of 3 phosphoglycerate. So here what actually is happening is the acceptance of this phosphate group from the first carbon position of this 1,3-bisphosphoglycerate. The phosphate group from the first carbon position of this 1,3-bisphosphoglycerate is being transferred to the ADP molecule and thereby regenerating 2 ATP molecules. So here as we can see that this 1,3-bisphosphoglycerate is transferring one phosphate group to this ATP and thereby leading to the production of 2 ATP. So this is called as the substrate level phosphorylation, because there is that involves the production of ATP from the given substrate or at the substrate level. So that is why we are calling it as substrate level phosphorylation. and the product is 3-phosphoglycerate. This 3-phosphoglycerate will again be isomerized to produce 2-phosphoglycerate, and the reaction will be catalyzed by phosphoglycerate mutase enzyme. Into the 9th step, which is the penultimate step, there will be a dehydration. Into this reaction number 9, each phosphoglycerate undergoes dehydration to give 2 high energy molecules of phosphoenol pyruvate, and the enzyme involved is this enolase. Enolase is the enzyme which catalyzes this reaction, which involves the removal of water molecule and conversion of 2-phosphoglycerate to phosphoenol pyruvate compound, which is an enol group containing compound. And the ultimate step involves the conversion of this phosphoenol pyruvate to pyruvate. Okay, so here what is happening is that the phosphate group, this phosphate group from this phosphoenol pyruvate is being transferred to the ADP molecules. Here this ADP is accepting this phosphate group and being converted to 2-ADP. Okay, ultimately producing pyruvate. So it is another step which involves the substrate level phosphorylation, and the reaction is being catalyzed by pyruvate kinase. Okay, so this is the pathway through which glycolysis is converting this glucose to pyruvate or pyruvic acid. So this is the overall reaction. This is the overview of the overall reaction. What is happening into the glycolysis? In glycolysis, 2 ATPs add phosphate to glucose and fructose 6-phosphate, and 4 ATPs are formed. These 4 ATPs are formed into this pair, in which the reaction number 7 is giving us 2 ATPs, and reaction number 10 is giving us another 2 ATP. So overall we are getting a net gain of 2 ATP and 2 NADH. Here the NADH is being formed at the reaction number 6. So this is the overall view of this overview of this process, where the glucose is converted to pyruvate and with the release of 2 ATP and 2 NADH. Okay, 2 ATP will be generated through glycolysis, and 2 NADH will again be generating 5 ATPs. So overall in glycolysis we are getting a gain of 5 ATPs.